Recording.
Okay, yeah, you can start.
Good afternoon, everybody. This presentation is about. I try. This presentation is about Geant4-DNA for flash radiotherapy, biological damage protections. My presentation is divided into two main parts in the flash RT mechanism and simulation part. At the first, I will try briefly explain about the flash mechanism and oxygen depletion as a main hypothesis in the flash effects. And then in the simulation part, I will look at to the simulate the oxygen depletion by Geant4-DNA. Let me start with the some general information about the flash effects. As we know, the flash is ultra high dose radiations. The in vivo experimental evidence showed the significant normal tissue sparing of normal tissue in this kind of irradiations. In this modality, in this kind of radiations, it means can permit the delivery the significant dose to the tumor, because this caused increase to the therapeutic window. Therapeutic window means when the normal tissue complication probability is decreased, we can deliver more dose to the tumor. Okay, there are different parameters related to the pulse flash delivery, but most of important of them is dose per pulse, total dose, total delivery time, and mean dose rates, which you can see the thresholds of these parameters are listed in the table. At this time, the flash mechanisms are not clarified yet, but various hypotheses have been proposed, but the main of them is the oxygen depletion. For example, the hypothesis about the reactive oxygen species is cause the less toxicity in flash radiations, or related to the immunological response. You can see in this figure. You can in the short time of flash irradiation, the smaller volume of the mouse. This is work? No.
But if you move it's not the mouse. If you move the touchpad, yeah. No.
The full.
The top left.
I can't see. This is this.
No, just if you go here. No. Put it here. So here.
It's okay. It's good.
Let's continue.
Okay. In this figure, you can see in the flash radiation, the short time of irradiation, a smaller volume of blood exposed, so less lymphocytes exposed to radiations, and as a result, we have preserved immune response. But I said about the main hypothesis related to flash effects is related to the oxygen depletion. So in the next slides, I want discuss about the oxygen effects. Okay, On the nanoscopic level, the oxygen effect is described as a combination of two main phenomena, which is strongly related to the chemical stages and indirect damage. So in our simulation, we focus on the chemical stages and focus on the chemical stage. We explain in the next slides, but related to the oxygen, the first phenomena is oxygen fixation hypothesis. It's related to the cellular damage. It means when in the presence of oxygen molecules, the damage are unrepairable. And then the next one is related to generation of toxic reactive oxygen species. According to these reactions, the radiosensitivity will be increased. This increased radiosensitivity related to these two main phenomena can be quantified by index of OER. OER means oxygen enhancement ratio, the ratio between corresponding doses related required to the different level of oxygenations to obtain same biological effects. This index is important to us because with this index we can quantify the radiosensitivity in persons of oxygens. Okay, I said about the main hypothesis, oxygen depletion. What is the oxygen depletions? According to these reactions, you can see here, radiation can consume locally oxygen molecules. So in the short radiation time in flash, according to these articles, because these articles is related to the oxygen diffusion into mammalian cells, the reoxygenation from the nearby capillary is the order of the milliseconds, but the order of the time radiation in flash is the microseconds. So by consuming of the oxygen according to these reactions and this short time, we don't have any reoxygenations from capillary to the target. So we have no reoxygenations. This is called oxygen consumption or oxygen depletions. The question is related to the oxygen and to the flash effects. Another question is, okay, why is the flash effects so differential? It means it's happening only in the normal tissue, not in the tumor. The answer to these questions is not clear yet, but maybe it's related to the oxygen depletions. It means, as you can see in this graph, in the normal tissue, in the physical normal tissue, there are large rapid changes in oxygen concentration. So radiation resistance increase, and as a result, we have protection of normal tissue. While in hypoxic tumor, the changes in oxygen concentrations is very small. So we don't have any significant changes in the radiation resistance. Okay, these results can be showed, or maybe it's better to explain this phenomena by OER. It means the depletion of oxygen-induced radiobiological radioresistance through OER, and this effects, this differential effects maybe can be explained based on the difference OER for normal tissue, which is larger than difference of OER in tumor. Okay, at this moment, there are some limitations, and because of these limitations, maybe the mechanism of flash is not clarified yet. The first of them is related to the sparing effects. Okay, sparing effects has been demonstrated only in vivo experiments, while in vitro show no significant normal tissue effects in flash regimes. It's maybe related to the conventional equipments in clinical routines. It means it's not possible to exploit conventional studies to model a flash. And it's related to the standard monitoring and dosimetry techniques in clinical routines, which needs to the sufficiently high time resolutions. And this time resolutions is not possible to the conventional equipment in the beam delivery or everything's in the usage in the clinical routines. And the most important of them is related to the ion recombinations. Ion recombinations is very significant for affecting to the ionization chambers, and it caused to limit the answer and to the accurate answer of this equipments in the flash regimes. And also, there are some limitations related to simulations of flash by Geant4-DNA, or it's better to say we face to this limitation in our simulations. The first of all, and the most important of them, is related to the, currently Geant4 does not have ability to simulate the dose rates. So for the solutions, we simulate the effect of flash indirectly by post-processing. And it caused maybe our results not very accurate. The second of that is related to the, in the Geant4-DNA, in the setup of libraries, which can simulate just biological materials such as water and DNA. We want to investigate the oxygen related oxygen concentrations. So it's need to add the extra material to this available materials. So for solutions for solve these problems, actually, we add the oxygen in the external file. You can see external file in the here. This external file just have effect on chemical stage. It means in the physical stage we have just water, and in the chemical we have water plus oxygen concentrations and effect just on the chemicals. And the last one, or the last limitation we faced with the simulation of oxygen diffusion rates, because we want to investigate the oxygen concentration and calculate of oxygen concentration. So it's important for us, we can simulate of diffusion oxygen from air to the water. And for the solutions, we assume the oxygen molecules is a background. It means the diffusion rate is 0. And for showing these features, we defined a specific reaction types. You can see here the reaction type of 6, specifically defined for oxygen interaction with other chemical species. You can see here. And this type of interactions defined as a, according to the diffusion rates. It's important for us, this type of interactions show us the oxygen considered as a background with diffusion rate 0, with special features. As I said in the beginning, I said we focus on the chemical stage in our simulations. It means in this simulation, at first, we calculated time-dependent G values of different chemical species, especially oxygen-related species. What is G value? G value, okay, and the other slides, the number of chemical species which produce or lost after irradiations per 100 electron volt energy of depositions, okay? After calculation of the G value, we tried with post-processing, calculated the oxygen consumptions and OER weighted dose under different dynamic oxygenations. And to evaluate our results, we compare our results with the tracks cam, which done by Boskolos et al. This slide is related to the geometrical setups for our simulations. You can see here we are considered two differential setup for electron and proton irradiations, because proton and electron have a different behavior. It means, for example, the electrons deposit its energies very sparsely by multiple scattering. So we must consider a big volume for stop all of secondary and primary particles inside the volume. But this vast volume is need to a lot of CPU time for simulation. So for solving these problems, we consider track segments. Which is track segments? Track segments is the thresholds of energy depositions. You can see here, we consider in our simulations, the amount of this energy thresholds is according to the, it's actually is trade-off between CPU time and generation of proper number of chemical species, because we want to calculate the G values. Also, the range of this kind of energy in electron in water, the range of this energy is comparable with dimension of cell nucleus. It's okay for us because we want to consider in the cell nucleus is important for us. But for example, the proton deposit all of energy in the small box. So we consider a small box, you can see here, for the proton. And especially for the simulation of flash conditions, we considered irradiated water box is sealed. It means isolated from oxygens. So it's not possible to reoxygenation from outside to inside the targets, just that for the simulation. But the irradiation particles and another setups is conventional. Just consider this condition for the flash. You can see in these slides our results with Geant4-DNA. You can see in this plots, the plot is time-dependent G values of chemical species produced by 90 million electron volt at different oxygen, under different oxygen concentration. You can see with increase in the oxygen concentrations, the electron aqueous, the black plot, and the hydrogens in the blue. The consumption of this is increased. It means, okay, according to these reactions, all of oxygen, all of available oxygens in the target can consumed with these two radicals. And in another plots, you can see the proportional of the reduction of electron outputs and atomic hydrogen. You can see, okay, the increase of the superoxide anion and hydrogenated form. Okay, in this situation, the radiation of electrons at different oxygen concentration, and then you can see the comparison with tracks. And in this slides, you can see oxygen concentration as a function of dose under flash for million electron volt. In this plots, 7 and 5% shows the physics normal tissue, and 1% indicates the hypoxic tumors. As you can see, for all of initial oxygen concentration, you can't see any oxygen depletions and oxygen reductions not observed in the range of clinical range, because clinical range between 20 until 30 in gray, but you can see, okay, it's a more, and I can show you in the next slides, for example, more than, for example, 120 degree, we can oxygen consumption, not complete oxygen consumption. Okay, in this slides, we can try to calculate the oxygen consumptions or different oxygen concentrations as a function of dose. It means as a more dose cause a more production of chemical species, especially oxygen species. And this means the more species has a results, the more consumption of oxygen, okay? When oxygen is decreased, the OER show radiosensitivity has a lower amount. It means the radiosensitivity with the more dose and more oxygen consumption is more lower. So for the calculation, the oxygen effects, we calculate OER weighted dose under dynamic oxygenation. It means after irradiation of flash, the oxygen concentration has changed. But in conventional, the oxygen concentration is constant. So the ratio of these two parameters as a function of dose can be used to estimate the sparing effect achievable in flash conditions. In this plots, we try to show the estimate the oxygen depletion. You can see, for example, for 7 and 5% in the show normal tissue, you can see very negligible changes in this ratio. And for 100 in the black one, you can see the more changes or more consumption in oxygens here, but not completely. And in the high dose, not in the clinical range. As a conclusions, okay, by Geant4-DNA and OER index, oxygen depletion is not observed in the range of clinical dose, and complete depletions is not supported by this kind of simulations. The sparing effect is more important in tumor than the normal tissue. And according to these results, we can conclude the Geant4-DNA is a suitable toolkit for the flash, but it's need to further improvement, maybe in the time of simulations or chemical stage or considering the chemical interactions. And the last one, maybe the OER index is not suitable options for our investigating the oxygen depletions. This result is presented in the flash conference by Seiko, and this good results, the OER is maybe is not good index for simulating the oxygen depletion in flash regimes. Okay.
Okay, thank you. 30 minutes, perfect. Perfect sound.
Good.
There is someone connected or...
For what?
Yes, I'm here. Ciao.
Ah, ciao, Masha. No, there is some question. Okay, because the conclusion of this is that the point you, the sparing effect of flash radiation is more important in tumor tissue. So this can be seen from the Geant4 simulation, no?
Yes, because in this plot. Oh. In these plots.
In this plot, yes, you can. Okay, where is the normal tissue here? And
okay, the normal tissue is 7 and 5%.
Okay, around the top. So
this one is the tumor. You can see again more changes in this plot.
Okay, okay. So this is an indication, but this is not sufficient to say that OR is the responsible for
yes,
for this.
It's not clear. Okay, our simulation is not accurate, or the flash mechanism related to oxygen depletion.
And my question is, if you go in the last slide, which are so today the last one, the conclusion. So today, which is if you have to continue to study the flash effect understanding with the simulation, what in your opinion now we should investigate? Which is the next step?
Yes, I think in the perspective and in the next step can be consider the time, because we consider the time microseconds for the equivalent of chemical stages. Maybe it means the microseconds, the conversion of heterogeneous chemical to homogeneous chemical. Okay, but we don't know, is it correct? Is it the exact time considered for this or not? This is the time, and this is the maybe change the OER, because we don't know OER is a good options for the oxygen depletion or not.
But there are other authors that are studying because you say that Joao Seco suggested it, but what he said, there is some other things that we may investigate as a different index.
At the moment, I don't have any answer for these questions, because I forgot, because it's a paper of this one is published in 2022.
Yeah.
And I don't have enough time for the read all of.
But the community is looking for some other possibility, which are the. I mean, the oxygen depletion is one. There are other explanation for flash. Or?
It's interesting things in the flash conference is the most of the time is the discussion about, okay, oxygen depletion is the main hypothesis. Is it correct or not?
Okay.
the 5% of populations, yes, say yes. The half of populations say no.
Okay.
But this time, it's not clarified. Oxygen depletion
But
is a good idea.
okay, but there are
not.
other things that are in discussion apart the oxygen depletion. Other idea,
Other ideas related to biological accepts. For example, in these slides, for example, in these slides, you can see in the short time flash irradiations, a smaller volume of blood has radiations. So immunological effects may be effect on the
okay. Question, yeah.
Okay,
closer because
yes. It's more a curiosity than a question itself, but you said that there is this kind of debate on what is the main effect. But in your slides, I think it's slide 6.
See?
Okay, here. You can only do in vivo experiment because we miss the detectors, the technology to study dose and dose rate with a sufficiently high time resolution.
Exactly.
So is there a part of the community that is working towards filling this gap? Because I think that if we want to remain, of course, only in the field of simulations, we can make all the good hypotheses that we want. But if we want some real, let me pass this term, easy data, it's way easier to go in vitro and then start to develop here technologically. So is there a way to, is there an effort that is being made to make these, to fill this technological gap?
Yes, some studies is related, for example, the laser plasma for the delivery of the beam for the ultra high dose is very effective on our results because now we produce the ultra high dose radiation with conventional pulse radiations, yes? But if improve, for example, the acceleration, the dosimetry, I can say, okay, in the ultra high dose irradiation, we have a lot of ion recombinations, yeah? And this limitations is effect on chambers for measuring the absorbed dose. If improved this in instruments for flash regime, of course, we have a better results for for example, even our simulations. Yes. Yeah. The detectors, the accelerators, everything is maybe.
Yeah, there is this big technological gap in this field that will really boost the research.
But in biological, maybe its improvement will be better because in the related to the, I don't know, I'm not expert in biology, but it's related to the biology. It's better to investigate the flash effects. What's happening in the body?
Yeah, because this is most probably more biological than physics. It's possible.
But Pablo, related to your questions, in the next steps, in the TRAN groups, they try to simulate those rates with Geant4-DNA.
Exactly, because the Sorghia is working with this. In fact, to simulate in Geant4
those rates.
The effect of the very high dose rate.
But I can't understand. Maybe it's necessary to more study related to
yeah, sure, because I think that the point is that you simulate a lot of rays, or I mean, you produce a lot of free radicals. And then there are in other tracks other free radicals that can interact in the time of the
Yes, and ion combination is very important in this.
and the
because-
combination. And this is not present in
But
Geant4.
in Geant4, the first one.
Then
should-
everything else.
Yes,
that stuff,
and
yeah,
the second one.
Exactly.
It's
This is
maybe
something
the
that we think
synchronic
is,
system.
since a long time. Yes, this is probably one of them, and see what happens from the also at the chemical level when you have all these free radicals that.
Yes, very important.
Okay, Fatima, other question? No? There are questions from people outside?
No?
Possibile?
Okay, so thank you. Thank you, Fatima.
Thank you. Thank you,
So-
everybody.
Thank you.
So in 1 month we will have the other the other. Anyway. Ciao.
Ciao.
Ciao. Grazie, ciao.
Ciao. Ma scusa, io c'ho una domanda se è possibile.
Sì,
That's problem.
Yes, yes, Fatima, this is a question.
Bene, buonasera a tutti. Grazie per Miss Fatima Farokhi. So I have one question, if it's possible. You mentioned in about your presentation for electron you fixed 1 mega electron volt, and for proton 5.90 mega electron volt. Okay, can we use a different range of energy for electron and proton, and we use it for flash radiotherapy?
I don't know I understand your questions or not. Your question is why this energy is considered for the electron and proton? Yes.
No, my question, if it's possible, we change this range of energy for this radiotherapy.
Yes, we can change, but we want the investigate the effects of radiations inside the cell nucleus, because
Okay.
important that all of energy depositions happen inside the cell nucleus. So
Because we have a different dimension of cell nucleus for every.
yes.
Yeah, but Clara, it is possible to change the energy, of course. But
Okay.
I think that also the reason is connected because we make a direct comparison with the tracks. So we use
Oh, yes, yes,
we
yes,
used
yes.
exactly one energy for electron and proton that was used in the paper that Fatima showed. So in order to compare our results in Geant4-DNA with the tracks, we made exactly the same physics configuration.
Yes,
No?
and tracks choose this energy so for the comparison with the experimental data.
Esatto, because there were also some experimental, but in principle, yes, of course, we can change.
Yes. But the most important factor, the cell nucleus. The cell nucleus is related to the micrometers.
Yeah, of course.
So
Of course.
we
If you
choose
want
the
the particle stop there, yeah.
But
Thank you so much.
yeah.
Okay. So ciao, have a nice weekend.
Ciao.
Ciao, buon weekend. Ciao.
Ciao. Ciao, buon fine settimana.